Hello everyone, my name is Dhruvi Patel and I am a senior graduating this May 2020. The topic of my research is pathology and advancements of the Ebola virus, generally discussing about the biology behind the virus. Viruses are like sneaky hijackers invading normal living cells inside a host by proliferating and producing other viruses like themselves. The Ebola virus is a perfect example for this. It is also known as the Ebola hemorrhagic fever. The structure of the Ebola virus consists of nucleoprotein, glycoprotein 1 and 2, viral proteins 24, 35, 30, 40, and L, which is RNA-dependent RNA polymerase. GP1 is responsible for attachment and GP2 is responsible for fusion. The viral genomes have the functions of replication, attachment, inhibition, transcription, and budding. The incubation period is 2 to 21 days. Symptoms like fever, headache, malaise, diarrhea, vomiting, bleeding of organs, shock, coma, and eventually to death occurs in extreme cases. The causative agent is Filoviridae and its genetic material is single-stranded negative RNA. Pathogenesis of the Ebola virus occurs from the virus entering the host body from the animal reservoirs, affecting the immune system and causing death.
Hi, my name is Eric Delgado, and I am a Kean University graduate. Ebola targets monocytes, macrophages, and dendritic cells upon entering the body. It enters by a process known as macropinocytosis, which is a regulated form of endocytosis. When the nucleocapsid enters the cytoplasm, the virus's genome transcribes the positive sense messenger RNAs that synthesize into viral proteins. When the virus's machinery begins the replication process, the negative sense genome transcribes into a full-length positive sense RNA strand, which is used as a template to produce new negative sense RNA genomes for new virus. This virus is very sneaky. A few of the viral proteins inhibit the production of interferons, which are a type of cytokine, a protein produced by the innate immune cells that act as alarm bells for stimulating an immune response. They also disable the ability to communicate with our adaptive immune system as the virus spreads to lymph nodes. However, macrophages and monocytes can still produce other types of cytokines with pro-inflammatory properties and also have the ability to recruit other nearby white blood cells. This induces a cytokine storm. The virus spreads rapidly, hijacking the immune system, recruiting and infecting new cells, releasing cytokines in an excessive amount, and causing blood vessels in infected regions to become more permeable. This leads to bleeding and hemorrhagic fever.
My name is Peter Nesser, and I will be graduating during the spring of 2020. We must reintroduce ourselves to the 6 different types of species of the Ebola virus and understand that there is no cure but vaccination for pre-exposure. Starting with the Zaire Ebola virus, a live vaccine named Ervable was introduced. And for those who do not know what a live vaccine is, it contains that specific virus but engineered to the point where it does not affect the patient, but once it is introduced into the body, the immune system starts to attack it and wins, leaving memory cells to reattack the virus at a faster rate if the patient is reintroduced to the virus. So in our example, a VSV containing a protein from the Zaire Ebola virus is introduced through a vaccination. Next is the Sudan Ebola virus. ZMapp is an antibody vaccine containing a 3 monoclonal antibody which attacks the virus at a different location and different techniques, 2 of which neutralizes the virus to not multiply or affect any further cells, and the third protects the cell from the virus infection. We now must know that not every species of the Ebola virus is dangerous. For example, Reston infects humans, but no illness or death are reported to this date. Typhorus infected 1 person but survived. Bombali has no evidence that it infects people. But here comes the question, is that even though Bundibugyo is as dangerous as the Zaire and Sudan Ebola virus, why is there no pre-exposure vaccination? It is treated like any species by giving them fluids and electrolytes, offering oxygen therapy, using medication, and treating other infections as they occur. As we see, the Ebola virus attacked and destroyed many lives and many homes in the African community, and to this day, we do not have a cure for it. We were very close to the Ebola virus reaching our homes, but were thankfully unharmed. We were scared, but not worried about the virus reaching us because it was an epidemic and not a pandemic. But looking at COVID-19, will we ever find a cure? Would it be non-curable as the Ebola virus? Did we only worry about the coronavirus just because it has reached our territory? Many questions of why the world didn't react to the Ebola virus just like they acted to the coronavirus. But we must understand that every life matters, whether it is 8 feet away or thousands of miles away.